My pleasure today to have with us Dr. Sonja Schrepfer and Dr. Per-Ola Carlsson joining us to talk about an update on the first-in-human study of hypoimmune islet cell transplantation in type 1 diabetes. It's really an exciting time for Sana Bio and for these two scientists. The Sugar Science has been following them for a few years now and has interviewed them separately earlier in our tenure together, and we've been very pleased to see them bring this to bear. It's an exciting development. Just a little bit about each of them. Dr. Sonja Schrepfer is at the forefront of STEM immunobiology, and she paves the way for a treatment of wide range of diseases, from supporting functional recovery of failing myocardium to the derivation of other cell types to treat diabetes, blindness, cancer, lung, neurodegenerative, and related diseases. And her work demonstrates that protecting transplanted cells from immune rejection is the key to unlocking the potential of regenerative medicine. And she's a professor at the Cedars-Sinai Medical Center in the Departments of Surgery and Regenerative Medicine, and a scientific founder and SVP, head of the hypoimmune platform of Sana Biotech Incorporated. Per-Ola Carlsson, MD, PhD, is a professor of medicinal cell biology at Uppsala University in Sweden. He also serves as a senior consultant in endocrinology and diabetology at Uppsala University Hospital and as a director of Uppsala Diabetes Center. His research focuses over the years have been on islet physiology, islet vascular biology, stem cell research, beta cell replacement, and early phase clinical cell therapy trials in type 1 diabetes. He's authored more than 200 peer-reviewed publications, and he's the PI of the presented first-in-human study of hypoimmune islet cell transplantation in type 1 diabetes. Welcome, both of you, and we're really excited to hear what you have to say, an update on how the patient is progressing. And also, if anyone has questions, please drop them in the chat and we will field them at the end of the talk. Thanks again for joining us.
Thank you, Monica, for your kind introduction. We could start by saying that this is a proof of concept study where we aim to transplant hypoimmune modified primary islet cells. And we have this far done it in one subject with type 1 diabetes. The cells are allogenic primary human cells obtained from a brain-dead cadaveric donor. We implanted them after genetic modification, the hip gene modification, into the forearm muscle of the subject. We did not use any immune suppression, and since this was a first-in-human safety study primarily, we used a low dose of cells that we administered. The study is at Uppsala University Hospital in Sweden with me as a PI. The measured outcomes that we looked for was primarily then safety, but then of course we were very interested to see if we could repeat the experimental findings by Sonja on immune evasion and see cell survival of the cells, and this in two scenarios, both as detectable C-peptide and also if we could detect the graft by MRI. So it's a single arm, it's an open label study. We have approval for 2 subjects and have this far transplanted the first subject. The ATMP we call UP421, which is then allogenic hip islet cells. And safety is primary endpoint, and we will aim for that we have no treatment-related adverse events or any serious adverse events at all. The secondary endpoints, as mentioned. The dose is low. It's only 25 to 80 million cells, which is a dose of about 5% that needed to reverse diabetes when used in the intraportal location. And the sponsor for the trial is then Uppsala University Hospital. So the details in this study was that we selected subjects that had optimal metabolic control, so they have very high A1C over 70 millimole per mole, which is about 9%. We also had as inclusion criteria that they needed to be between 30 to 45 years of age, and have been having debut of diabetes in childhood and have had diabetes for more than 5 years. In this case, we had an individual that got his diabetes back in 1987, who at transplantation was 42 years of age and had an A1C of 96 millimole per mole, which is 10.9%. For the islets that we use from transplantation, we retrieved them from a O-positive blood type donor. It was a male donor, and we have a rather high islet purity, about 86%, and he did not have diabetes, and of course, with an A1C of 42 millimole per mole. So these cells, these pancreas from the donor were collected. The islets were isolated at the Rudberg laboratory at Uppsala University Hospital. The cells were then sent to Oslo University Hospital in Norway, where the cells were dispersed and also then genetically modified according to the Sana protocol to generate hip islet cells. And these islet cells were then reclustered and released as an ATMP back to Uppsala University Hospital. And on December 2nd, we transplanted this subject, and the procedure took about 90 minutes. The cell injection took about 60 minutes because we took real care to disperse the cells carefully so that they would not be clustered in the same site. And that also meant that we did have to make about 17 channels to distribute the cells. And here we have a short video actually of the implantation that shows one of these injections where the cannula is inserted and then slowly retrieved while the cells are infused or injected in this site. Then we took care so that the cells would not fleet out in the end of the injection. So the endpoints were primarily safety. We looked at cell survival and function with C-peptide measurements, graft visibility by MRI. We looked with Sonja's techniques for adaptive immune evasion and innate immune evasion. And as of 4 weeks, when the first readout was collected, there was no SAE yet in the study, and there has been no adverse events possibly or probably related to the study drug in the study. And we are continuously, of course, following this individual every second week at this time, and the next more thorough readout will be at 12 weeks. But we continuously follow him, and here are the data with regard to C-peptide at 4 weeks. As you can see, in baseline, we did not have any measurable C-peptide. It was below 0.48 picomole per liter. So it was not measurable at all by the used technique, whereas in the subject after transplantation, both at day 7, day 14, day 21, and day 28, we had about 10 picomole per liter of C-peptide, and it seemed to be a stable level over these 4 weeks. Just for comparison, you can make it ×3 to make it into nanogram per milliliter if you're more used to that. So here is also a mixed meal tolerance test that was conducted after 4 weeks. And as you can see, it started about 10 then picomole per liter at baseline at fasting. And after doing this mixed meal tolerance test, he increased up to 17 picomole per liter. So there was also responsiveness in the islet cells that we transplanted that we could see after 4 weeks. In comparison, the gray line at the bottom is a mixed meal tolerance test performed prior to transplantation, and then we did not have any measurable C-peptide at any time point. And the detection limit is then 0.48 picomole per liter. So these are also the MRI scans that were performed. We performed it weekly here at week 1, week 2, and week 4. And to start with, there was some fluid in the muscle around the injection site, but that disappeared rather quickly after implantation. At day 28, remained punctual signals that we could see by the T2 stir wait sequence, which is mainly fluid then, and we believe this is a high blood perfusion of the graft, but of course it can't be proved. But these are these red arrows pointing to these signals that are detected in the muscle after injection. We see no inflammation, and we of course also monitor by MRI for safety and see no adverse events in the muscle at the present stage. I'll leave a word to Sonja, please.
Thank you, Per-Ola. Yeah, I will show you how the patient's immune system reacted to the transplant. So as Per-Ola already told you, the hypoimmune islets, of course, got transplanted, but there were also partially edited islets in the drug product because we are not creating a clone. So it's more like a bulk population. So that means that what you see on the left side, the patient also received islet cells that we call WT or wild type. That means this is a normal donor islet cell, so with the HLA molecules of the donor. And then CD47 is not overexpressed. And we also transplanted islet cells that had removed the HLA molecules, but not sufficient CD47 overexpression. And then the hypoimmune cells, by definition, have to have the HLA knockout and the CD47 overexpression. So when you transplant that into the patient and you do blood draws over time, you can now look at the patient's different immune populations, like the T cells, the natural killer cells. You could also look for the antibodies the patient produces, and you can take those immune cells and put them on those subpopulations of the donor drug product. So for those assays, what we did is that we saved some of the donor cells that went into the patient, and we collected the blood of the patient. And what you see on the next slide is when we do this immune analysis. Next slide, please. So this is actually an advantage that we have a drug product that has those subpopulations, because what we can show you now here, this is the patient's T cells. I start on the left. So the patient's T cells are isolated baseline. That means before the islet transplantation, and then day 7, 14, 21, and 28 days after the islet transplantation. And what you can clearly see is that the patient's T cells recognize the wild-type islet cells. And that is because the wild-type islet cells do have the donor HLA. So those T cells do see the 4-ine allogenic HLA because the HLA reacts like a 4-ine allogenic fingerprint. So you clearly see that the patient's immune system, patient was not immunosuppressed, reacts as expected and recognizes an allogenic donor cell. That leads to killing of wild-type islet cells, as you can see on the left bottom. So whenever we see the T cell activation, that led to the wild-type islet cell killing. When we remove the HLA in the double knockout group in the middle or the hypoimmune cells on the right, you do see that the same T cells from the same patient are not activated when we run the assays against those subpopulations, and it does not translate into killing. You see that on the bottom. So that shows you when you remove the donor HLA, the patient's T cells don't see those cells anymore, and they are not activated, they're not killing. So on the next slide, you see when a T cell is activated, the T cell activates B cells, and then plasma cells produce antibodies, usually against donor cells when we do transplantation. And that's what you see on the left column. So what happened is that the patient's B cells or plasma cells produce donor-specific antibodies, first at the IgM. So you see the highest blue graph at day 7, and then you see the IgM antibody switch to IgG around like 2 weeks. You see that switch occur. So really like textbook immunology. And that shows you that the patient reacted as expected to an allogenic transplantation. What you also see is that the very same blood of the patient containing those donor-specific antibodies that recognize the donor HLA, those antibodies are not binding when you have an HLA knockout cell. So middle column or the right column with the hypoimmune cells, there is no donor-specific antibody binding, although we do know that the antibodies are there. And that is really the principle of immune evasion we are going for. Even if the patient has antibodies, the product, the islet cells, the hypoimmune islet cells should evade that recognition. They shouldn't bind. Then on the next slide, what we love to do is, instead of just showing that the antibodies are binding, we also love to understand the consequences. So when a donor-specific antibody is binding to the wild-type islet cells, it will activate the complement in the patient. And then you see complement-dependent cytotoxicity, or CDC, because the complement basically makes holes into the islet cells and would kill them. And at the same time, what you see on the right side, when an antibody binds, a donor-specific antibody, to the allogenic cell, it can also activate innate immune cells like NK cells. And then those NK cells would trigger the killing of the islet cells. That is called ADCC, so antibody-dependent cellular cytotoxicity. So we always love to study that in the patient as well. And that's what you see on the next slide. I start with a CDC, so complement-dependent assays. And what we did here is you have from the patient the serum that contains the developed antibody plus the complement. And you put that in the left column on the wild-type islet cells. And because I showed you that there are donor-specific antibodies are binding to the wild-type islet cells, that leads to complement-dependent killing. So you see in the killing assay, killing at all time points. When we do that for double knockout or hypoimmune, because no antibody was binding, I showed you that in the flow analysis, you see subsequently there is no CDC killing. And then for the ADCC data on the next slide, you see when we do the serum of the patient with the antibodies, you decomplement it. So no complement, but now you're adding the patient's NK cells. When the antibody is binding to wild-type islet cells in the left column, that leads to ADCC killing of a wild-type islet cell. But what you also see is if there's no antibody binding, middle column, and on the right for hypoimmune, no ADCC killing. So just highlighting the fact of that immune evasion concept. Then on the next slide, you see the innate assays, because so far the middle column and the right column, they look the same. So to overcome the adaptive immunity, it's really the HLA knockout. But because we are creating now a cell with no HLA called missing self, innate immune cells are killing missing self cells. And that's what you see in the middle column. The patient's NK cells are killing a double knockout islet cell immediately at any time points. But because hypoimmune overexpresses the CD47, you see in the right column how the NK cells are not attacking the hypoimmune islet cells, and they can overcome also that innate immune barrier in the transplantation setting. We always ask ourselves, are we missing something? So the immune assays I showed you so far, we selected the immune cells from the patient. So we separated the T cells from the NK cells to run the assays. But it could be that those immune cells need communication with each other to really kill. And that's why we do this, what I call the killer experiment, where we have from the patient everything we can get with a blood draw. That means the PBMCs containing T cells, NK cells, etc., and then the serum containing the antibodies and the complement. And we put everything on the subpopulations of the drug product. And what you see on the left side is wild-type islet cells do get killed by the patient's immune system. The double knockout islet cells in the middle do get killed. But when you do the experiment with the hypoimmune islet subpopulations that were transplanted into the patient, you see that there is no killing. And because those graphs, they sometimes look boring, we thought we show you a video. So the video actually shows you exactly what I showed you on the graph. You will see in green the islet cells, and then we put the patient's PBMCs and serum on top of it. And what you see with your own eyes is that a wild-type islet on the left gets killed because of the adaptive immunity. The middle column, HLA knockout islet cells get killed, innate immunity, and you see that is the drug product, the hypoimmune islet cells with the patient's immune cells, and you see in green how beautifully they survive. So there is no killing of the hypoimmune islet cell population by the patient's immune system. And with that, I hand over to Per-Ola. Thank you, Per-Ola.
Yes. Thank you, Sonja. And so this, from our side, has been an amazing collaboration with a lot of individuals involved, of course, both at Uppsala University Hospital, that in Swedish is called Akademiska sjukhuset, and then, of course, Oslo University Hospital, Oslo Universitetssjukhus, and Sana Biotechnology that brings in the technology. And the study has been funded by the Leona and Harry B. Helmsley Charitable Trust. And of course, I would say that this brings great hope for individuals with type 1 diabetes. We should say that in this first individual, we transplanted only a minute amount of cells, about 5% of a curing dose. So we did not have any major metabolic effect in this first individual, but it hopefully will provide proof of concept that the cells evade the immune system and that we will have long-term function. This far we describe it for 4 weeks. Thank you for your attention.
Thank you both very much for this great update. I have a question already here, a couple questions from the chat, and I'm going to ask them and then ask people to raise their hand and we can unmute them for more discussion. Peter Lemedico, pro-insulin to C-peptide ratio. Not sure where that.
We have this far not analyzed pro-insulin. We do have additional serum samples that we could add on this kind of analysis if wanted.
Ashley Buckle, did you phenotype the patient's NK cells?
So again, much more work will be done. At the moment, I can say no, we did not do that yet, but we are in the middle of that study. So in a publication or so, you could expect to see some of those data.
Jason Gaglia, are the measurements of C-peptide in the arm with the islets versus the other arm or systemic level?
It's systemic level, so it's collected from the other contralateral arm.
Great. Any other questions? Oh, Don Goldstein, do you know when the SC451 will be in clinical trials?
Yeah, so Sana announced that you will hear about it when we are 6 months before an IND filing. So therefore, I can unfortunately not give you a better timeline at this point. But if you watch their announcement, so 6 months before an IND, you will hear from Sana.
Great. Did you measure SERP alpha expression from Gabriel Circusano?
SERP alpha expression on the NK cells, we did not show that in the data, but you will see that in a publication, yes.
I'm curious to know if there are any concerns about volume restrictions in the intramuscular site when a therapeutic full dose is delivered, Chris Spencer.
Well, it's mainly a matter of a dispersion of cells, so we don't foresee any major problem with increasing the dose. We have transplanted previously much larger doses when doing it as an auto transplantation, actually, for individuals that have gotten a pancreatectomy. I know from the China experiment quite recently published, they also transplanted even a curing dose at the intramuscular site. So we foresee that it will be possible, but just dispersing the cells.
Nicole Rogers, I may have missed how you transplanted, but is it possible, did you transplant the different cells, wild type, DKO, HIP in the same place? If yes, would you expect a negative impact on the HIP cells due to the death of the wild type cells?
So we wouldn't, we have studied that with spiking experiments in the preclinical data package. So we know that the targeted allogenic rejection process is specific enough that if you even have them at a similar location, that hypoimmune can survive. We had done the experiments in mice and in non-human primates, and we do see it now in the patient as well, that there is no, it doesn't seem to harm hypoimmune.
Alex Drilling, thank you to everyone involved. Can you describe next steps? Example, when will the study population size increase, and will this work with manufactured cells? For example, those made by Vertex, the VX-880.
The plan will be to go into stem cell derived insulin producing cell product, and then of course increase the doses. So that is more or less the next step that we foresee.
Okay, great. I'm sorry, there's so many questions. Chris Clifford, will the graft be studied or monitored for any viral or bacterial infection? That's key.
Well, we can't, of course, we will not do any biopsies, at least we have not planned to do it. But we, of course, have strict release criteria from the manufacturing unit so that we screen the cell product, of course, for any viral and bacterial infections in the product that is released.
Julia Parnas, how are the manipulations done to the islets affect islet cells' longevity and activity?
Hopefully not. But the editing process is harsh on the islets. You're losing a lot of cells during the manufacturing process, etc. It is an editing process and an lentiviral overexpression. So it's really, it's a harsh process. Manufacturing is 1 week. So you're losing islet cells on that way. I do hope that the ones we transplanted themselves, because they are viable, that the longevity will not be impacted. But I guess that has to be seen in studies.
I think the major challenge for us will be in this subject that historically has had a poor metabolic control, but we might foresee some glucotoxicity over time. So that might limit function, but hopefully we will manage to keep him in much better metabolic control than historically.
This is my question, regarding the safety switch that you use, the anti-CD4 IgG4, in your non-human primate studies, have you guys been considering a safety switch in this context with this human patient?
I mean,
Well, there are, of course, that possibility, but it's not an approved drug product at the present time, so it's not in the protocol or anything. And that is also, of course, a reason to strictly monitor the site and also have a possibility to retrieve the graft even manually, since we implant it intramuscularly.
Right. Manish Jain, what is the max volume you anticipate for intramuscular injection? Will it require multiple sites of injection?
We will reasonably need to spread it out in a larger muscle area and perhaps also use both arms. But it is possible to inject this number of cells if we just spread them out. So in this case, we only have one incision hole, as you saw, and used the injections upwards or downwards in the arm. So you could, of course, increase the area tremendously.
Arkan Gusani, can you please elaborate about the AE/SAE that the patient experienced, since it was not related to study drug? Was it due to underlying medical history or occurred post-treatment?
We have not had any SAE at all in the study. So I think that was a misunderstanding, sorry.
Okay, great. Can you speak to the issue? This is Alexis Karma. Can you speak to issues with the clinical trial, lessons learned so far about patient recruitment, procedural issues, and monitoring, etc.?
So in the protocol, we investigate subject weekly in these first 4 weeks, and then it has been bi-weekly, and then it will be prolonged. But it is mainly safety measures, and then it's quite many MRIs that will be conducting during this first year. And that is the major safety screens that we are performing. Otherwise, it's some functional studies that will be performed besides these MMTTs, which will be repeated a couple of times during the first year. We will also have a PET MRI investigation at 2 occasions.
Great. I just wanted to ask personally, could you discuss the injection site that you used, and did it affect any motility of that muscle? What are your thoughts about using the abdominal rectus sheath as used by Shushan Wang et al. In the 2024 Cell paper, where they also used chemically modified stem cell?
Absolutely. I think most muscles will be performing quite similarly. So in this case, we only have a minor number of cells. We implanted in the brachioradialis muscle. Perhaps it could be a problem if we would select a muscle which is very strong, like hamstrings or something like that. But otherwise, I think it's doable to select most muscle in the body.
Quimei, any genome instability seen in iPS-derived hip cells?
Cannot comment on that because that study is really cadaveric primary islet cells, so there is no issue here.
We had another question. You know, how can you hypothesize as to the immune response against partially edited cells? Oh, sorry, that's not the question I had for you. Can you expand on how you're going to produce a gene-modified master cell bank from your GMP-compliant PSC line that's genetically stable and remains so after your gene editing and differentiation into islet cells?
Yeah, so that will be the SC451 program. So it's not related to the primary islet study we did here. So you will hear about that in the future.
Okay. I didn't know if there was any kind of, intersection there. And then, like, once these cells are implanted, do you expect any of the modified cells to proliferate? And would the daughter cells maintain the gene edits?
So we studied if the donor cells have stable the gene edits, and that as long as we did the non-human primate experiments, which was many, many months, it stayed stable. If it wouldn't stay stable and they would lose the hypoimmune phenotype, we would lose the cells, obviously, but we have no indication that it wouldn't stay stable.
Kiadi Gerder, do you plan to study how immune cells interact with hip islet transplants during an infection? Are there any potential trade-offs between reducing immunogenicity and altering the physiological functions of islets, such as insulin secretion or glucose metabolism? And that's a good one.
So that we studied in detail in the regulatory package, etc. The 3 hypoimmune edits I described are not impacting the islet biology or function. And then if you, for example, have a bacterial infection, we studied that in mice when we infected the mice and had the hypoimmune islets, also nothing happened. So that is the principle of immune evasion. The immune system is allowed to be activated, etc., even against the donor HLA I showed you. And still, the edits should protect the cells. They shouldn't be seen by the immune system. And we would expect viral or bacterial infections that wouldn't happen either. So we did LPS, we used adrians and different bacteria in mice. We did publish that we have a hypoimmune mouse where you can see that the mice can reproduce, can live a normal life, basically, with those 3 edits. So we are not expecting that those edits change anything about the islet biology or anything else.
Great. Ye-Chun Po, is cryopreservation part of the process? If so, were they cryopreserved as single cells or islet clusters?
Oh, we did not use any cryopreservation, so we transported them actually by air from Oslo to Uppsala. Actually, for islet cells and islets as a whole, they seem to be damaged quite quickly by freezing, so we try to avoid that.
From Th1nk, are you concerned that lentivirus mediated gene editing would lead to safety issues such as tumorigenesis?
Well, the editing process, or there is certain risk, but for the CAR studies, most of the CAR constructs are using lentivirus, so we have a lot of long-term clinical data on that. And so far in our safety package also, we didn't see anything, but you always have certain risks when you do LVV or gene editing. So each technique, I think, has its own risks.
From Kristen Schutz, for clarification, you dispersed islet clusters previous to injection and injected into multiple pearl string channels. How many channels do you usually create for a full auto-transplant dose?
Actually, I think normally they are not as careful as we was this time. So normally I would say they use about 20 channels, but perhaps a little bit longer channels that we use today. We took real care in this time to disperse the islets, and perhaps more care than in clinical practice.
Great. Scott Karmer, product purity. Are there efforts to increase the purity of the final gene-edited islets to remove all but the hypoimmune edited islets? Is this necessary for clinical effectiveness? How difficult would it be?
It is difficult if you work with primary islets, what we showed you, but for the stem cell-derived islets in the future, you will do the editing in the stem cell state. You pick a clone, and then your whole drug product will have the hypoimmune edits. So what I showed you today is solved in the stem cell-derived islet field. It's really only for the primary islets. And there you saw the graft still can survive, but of course you're losing the unedited or partially edited cells. So your efficacy goes down, right? So that is the disadvantage of what I showed you today.
We'll take a few more. I feel like I'm peppering you guys with questions, and you're doing so great. Manish Jain, where they administered as aggregates, clusters, or any other support material used along with the cellular product?
There were no support material. It was only islet cells, but they were clustered so that they stick together as ordinary islets.
Sean Martin, are there plans to monitor this patient for oncological risk? If so, how often, and what does it entail?
So the major readout we have for safety is the MRI scans that is repeated. I don't know if it was about 6 times the first year, and then we will continue to monitor this subject for up to 15 years according to European regulations.
Great. Torsten Meissner, could you even get enough edited cells from one donor for a curative dose, or would you need to pool several donors?
Yes, you would certainly need to pool several donors, but also for islet transplantation without edits, like when you then use immunosuppression, you need 2 to 4 donors usually to get insulin independence. And you would need more with hypoimmune because we are losing islets with the manufacturing.
But as mentioned previously, this is proof of concept. So the way forward would be stem cell-derived insulin-producing cells, and then there will be no limit in this case.
Great. Ola Denske, thank you. Are you planning on presenting these data at a specific conference with follow-up data?
We do have an invitation to ADA, so we will be presenting there.
In June. Great. Alexis Carmer, how is the patient managing their diabetes? Automated insulin delivery or?
Actually, he's on insulin pens presently, but we are planning on putting him on an Omnipod 5 within soon.
Okay, we'll take a few more here. If islet from category is islet transplantation is needed, can beta cells alone be used for the transplant instead of whole islets? And what would you expect in terms of functionality?
I can only speculate. I think you need the islet cluster, the supporting cells. So for that reason, we started all the projects with the islet clusters, and we did not aim to only transplant the beta islet cells.
Aidan Levinsky, are minor histocompatibility barriers such as mitochondrial DNA polymorphisms in hip cells a challenge to hypoimmunogenicity?
Great question. And we published on that a few years ago with the mitochondrial SNPs. Yeah, they are not, short answer, because we have no HLA. So those SNPs will not be presented because the HLA is completely removed. That's why an MHC or HLA knockdown does not work in our hands. We have really to knock it out completely.
Okay. What prompted the arm as an implantation site?
We selected muscle that we had previously used for auto transplantation site. So that was, and perhaps the auto transplantation choice from the start was based on parathyroid experience in the past.
Jason Gaglia, what was the glycemic control at time of initial pre-transplant MMTT versus 1 month MMTT? Question is, around any chance of endogenous recovery with improvement in glycemic participating in a clinical trial?
Well, the A1C have decreased slightly, but not tremendously in this subject. I think it was about 63 millimole per mole, that is about 8% after 1 month, and it was about 10% to start with. So, yeah. So I wouldn't say that there is a chance of regain of function. This individual, we have investigated him before, and he has had no C-peptide for decades.
Mm-hmm. I did have one last question for you both. Who is on deck to reproduce these findings outside of Sana? Do you know? Have you thought, I mean, do you think that other groups are, now sort of catching up and trying to reproduce these? Or is there a chance that a collaborative effort could push things forward faster?
Yeah, so Monica, there are so many different ways how to think to overcome an allergenic barrier. You see so many colleagues working in that field. We show you one concept, what we call immune evasion. You can induce tolerance. So there are so many different strategies. So it's a really exciting time, so much going on in the field. So it's really fun to watch it. But this is not the only strategy we showed you today. This is our strategy, but you will see many other things moving forward by others, etc. So really exciting.
Yeah. Oh, no, there's definitely, the race is on, but I'm just curious as to, I wondered, I'm assuming that others are now working quickly to, reproduce these findings, and that will really give a lot of credence to this N of 1, right? To really push things forward faster and gain more funding and all the good things that are needed. All right, then. It's, we've had you for 45 minutes here, and we promised a half an hour. So thank you very much for your time and for fielding this, so many questions. It shows that this is a very exciting piece of research and that people are very interested in seeing it progress. So my best from the Sugar Science to you both, and we look forward to hearing what you have to say at the ADA, as well as any publications that are forthcoming. Thanks again, and have a great rest of your day.
Thank you.
Thank
Have a
you.
nice day. Bye.
Bye now.